Welcome to Plants in Python. My name is Bob Van Buren, and I'm an assistant professor in the Department of Horticulture at Michigan State University. And so in today's lesson, we're going to talk about genome visualization and comparative genomics in plants. So in the last lesson, we talked about genome assembly, and so we went from a raw sequence reads to an assembled genome. And this is really just a starting point. And so now we have this seemingly random set of A's, T's, C's, and G's. And the question is, how do we draw biologically meaningful data from this? And so today we're going to talk about the various ways that we annotate genomes, what genomes contain, and how we can analyze them. And so the plant biology-based learning objectives of today's lesson are to understand what makes up the genome and how genomes vary between plant species. And so plant genomes are dynamic. They vary incredibly in the gene content, genome size, and so we can use that information to discover biologically important concepts. And so we'll also introduce the file formats used to denote features in the genome and talk about how these files are created, how we can manipulate them, and how to understand what they're showing. We'll also generate a basic understanding of the foundational principles of comparative genomics, including Synteny and orthology-based approaches. So we'll talk about the different ways that we can compare genomes and how we can draw meaning from that. So genomes are filled with lots of different sequences with various functions. And so the thing that we typically care about the most are the genes. And so if we think back to the central dogma of molecular biology, we have here, this is a gene. And so a gene is made up of primarily exons and introns, and then also has some promoter sequences, some terminators, and some other sequences that we won't get into in too much detail here. But this DNA gets transcribed into RNA. This RNA gets spliced where the exons, this is what actually codes for the protein, get retained and the introns get cut out. This mRNA is then translated into proteins by ribosomes. These amino acids are folded together, they have post-translational modifications, and then we get proteins that serve all sorts of functions in the cell. And so typically these are the things that you would care about most in a genome. Maybe you're working with some molecular biology, trying to clone genes, maybe you're doing some comparative genomics or some breeding or something. But this is kind of the thing that most people care about. But in reality, genes only make between 1 and maybe 10% of a plant genome. And so there are a lot of other features that have biologically important roles. And so outside of genes, there are tons of other elements that have importance. And so for the last 20 years after we sequenced the human genome, the next question was, well, what's in the human genome and what is it doing? And so this is an ongoing project called the ENCODE project, which stands for the Encyclopedia of DNA Elements. And this was a project that aimed to identify the functional elements in the human genome. And so this project has been underway for decades. I don't know what the total cost is, probably in the order of billions. And similar projects have been undertaken for plants, so mostly maize and Arabidopsis, rice and a couple others, and some other model organisms as well. And so the goal of these projects is to utilize these various different sequencing approaches and different libraries, we won't talk about these, to identify different elements that are important in the genome. And so we have, of course, our genes here. This is kind of the central thing we care about. We have various promoter and enhancer type elements, and so these are sequences that transcription factors and other cellular machinery can bind to enhance where and when a gene is expressed, to stop it from being expressed, and to tell the plant, or in this case the human, when and where to express different things. We have how the DNA is packaged within the cell, and so how open it is, so if it's wrapped up in histones, or if it's in sort of this open chromatin where it's accessible to different transcriptional machinery. There are different histone modifications that can control if chromatin is open or closed. We have gene methylation, which can control what can bind there and if it's more likely to be open or closed. There are small RNAs that can degrade transcripts, that can enhance them. There are other non-coding RNAs. So there are tons of elements within the genome that have functions as well. And so in this course, we'll be mostly talking about the genes, but know that there are tens of thousands, hundreds of thousands, maybe even millions of sequences that are important as well. And we're really just scratching the surface here because even in the human genome, we don't know all the elements that are functional and the role that they're playing in disease, in gene regulation, and processes like that. So this is kind of an ongoing area of research, but as the deeper we dig into this, the more complicated it becomes. And so the other major factor or other major component of genomes are repetitive elements. And so these fall into two main classes, and most repetitive elements are transposons. And so these were pieces of DNA that likely originated from viruses, and they kind of replicate uncontrolled in the genomes of most living things. And so we can break these transposable elements into two classes, retrotransposons and DNA transposons. And so retrotransposons have a couple different proteins in them that allow them to basically transcribe their sequence into RNA, turn that RNA back into DNA, and then integrate somewhere else in the genome. And so retrotransposons are really good at doubling themselves, so they can duplicate and proliferate uncontrolled, and they make up the majority in general of transposons in plant genomes. And then we also have this other class, this class 2 transposons, and these are DNA-based. So these are unable to replicate themselves and duplicate somewhere else. They just typically will move around the genome, so they will cut themselves out and insert themselves somewhere else. And they're mainly duplicated by issues with uneven crossing over during meiosis and other issues like that. And so these elements are typically non-functional, although they do have some functions which we'll talk about, but they make up the majority of a lot of genomes. And so, for instance, in the human genome, almost half of our genome is made up of these repetitive elements, with the vast majority of them as, I think, these line elements. And so I think there are hundreds of thousands of these in the human genome. Some transposons are active, meaning that they are continually duplicating and moving around, and some are inactive and they're just kind of there and we can't easily purge them. Similarly, the mouse genome has about the same proportion. Drosophila and C. elegans have a lot less. And then the model plant Arabidopsis has only about 10 to 20% of its genome is composed of transposons. By comparison, the maize genome, which is considerably larger, so it's in the same order of magnitude as the human genome, about 80% of it is repetitive. And so it's filled with tons and tons of retrotransposons. And we typically think of these things as not important, but it turns out that these repetitive elements have a really important role in gene regulation, gene duplication, and other processes. And so a transposon can insert in front of a gene and change if it's being expressed or not expressed. And great examples of this in domestication are like grape color. So like the difference between red and white grapes is caused by different transposons in different varieties of grape. Fruit color and size and tomato and dozens of other domestication traits are caused by a transposon inserting and either interrupting or giving a new function to a gene. Repetitive elements can result in gene duplication. So when a retrotransposon duplicates itself, sometimes it will take a chunk of another gene or a whole gene itself, and it will put it somewhere else, and that new gene can take on a new function. And they can also serve to kind of rearrange genes and make new proteins in the genome. So they're kind of a nuisance. Plants and animals are continually trying to purge them and stop their proliferation, but they also give rise to evolutionary novelty. So it's sort of complicated, but this is a really large and important factor of plant genomes, and we're still trying to understand, again, what their role is and how many of them are functional. And so we've kind of talked about the different elements of the genome. So the question is, how do we format and visualize these different features? And so you could technically open up a genome FASTA file in a text editor or terminal, but again, it's just going to be A's, T's, and C's, and G's, and so humans are generally unable to identify meaningful sequences by eye alone. And the maize genome has like 2 to 3 billion bases. Like we can't really visually find anything there. And so we can use genome browsers, which we'll talk about a little bit today, to move around the genome and identify various tracks of biological features. So these could be genes, could be repeats, they could be non-coding RNAs, variants, and other biologically meaningful sequences. We can store these genome annotation features in various tab or comma delimited or delineated formats. And so there are two that we're going to talk about today, the bed file and the GFF3. And then you can use this in a lot of downstream programs for comparative genomics, for quantifying various elements in the genome. And this is kind of the central thing that we care about with formatting genome features. And so the first format that we'll talk about is the bed file, or the bed format. And so again, this is a comma or tab delimited format, and it consists of one line per feature, and each feature containing 3 to 12 columns of data plus optimal track definition lines. And so the first 3 that are essential in the bed format are the chromosome and the start and the end. So here's kind of an example of the simplest way we can have a bed format. So this would be chromosome 1 of whatever organism you're working on. This will say the base pair that that feature begins at. This is the base pair that it ends at. And the other columns of data would be something like maybe the gene name, the classification, is it a gene, is it a transposon, is it a promoter sequence, and then various features of is it on the plus or minus strand of the DNA, does it have a quality score, things like that. So this is one way of taking this massive sequence file and just delineating the most important features of the genome. And the other format, which is probably a little bit more common, is the GFF format, or the general feature format. And so this one is pretty similar to the bed, but a couple notable distinctions. And so GFF is a tab delimited file with 9 columns of data plus some optional tracks as well. And so we're not going to go through each of these, but basically we have this first line that denotes it's a GFF, and then we have this first column is going to be the chromosome or the contig name of the piece of DNA. The second will be a source, so this can be blank, which is the case here, or it could be a program that generated it, or some other feature that we might care about. The third column is the type, and so in this case, this is going to be an mRNA. So this is going to be the full length sequence of this protein, or in these cases, it's exons. These are the actual pieces of the protein that get, or the DNA that gets spliced out to make the protein. Then we have the start and the end within that sequence, and then various things like the strand, quality score, phase, and then this is another thing that we care about more, which is the actual name of the protein or the feature. And so in this case, we have this gene here, which is just called mRNA 0001. It starts at this position, base pair 1300, and ends at base pair 9000 in the genome. And then the exons go from here to here, right? So we have this first exon here that starts from base pair 1000 to 1500, this one which overlaps a little bit here, and then it goes all the way from 7000 to 9000. And then these are named exon 1 through 5. And so this is really, these are kind of the two workhorses that you'll see in genomics. There are a couple other ones, but there are various tools to translate between them, but oftentimes you'll need one of these two formats if you're going to do some comparative genomics or some downstream analyses. And so we can import these two files or into a genome browser, or you can just look up a genome browser for your favorite organism. So this is from the Arabidopsis genome. This is a JBrowser for Arabidopsis. And so it's a really nice GUI-based program, so we can just go to the website and we can move around to different features. And so here we're showing kind of just a random subset of chromosome 1 in Arabidopsis. So this is from base pair 21,000 to 39,000. We can see different features here. So the first one is going to be the gene locus. And so these are a couple genes in Arabidopsis. So we have this first one here, AT1G03993, another gene here, another gene here, another gene here. These are the protein coding features of those genes. So this is the actual locus itself. And then these are the intron exon structures. Sometimes we can have an alternative splicing event, like in this gene here, where we have 0.1 and 0.2, and you can see there are some notable differences. Or in this case here, this is even more significant, where we have, it looks like 8 different isoforms of this gene. So this transcript can start at this start codon. Actually, it's going in this orientation. So they can start here, they can start here, they can start here. And this just gives all the different transcripts that are available for that gene. And then we can also put on other features here. So in Arabidopsis, we have really great mutant libraries where they have transposons, TN5 transposons that have inserted into all kinds of different genes. And so these are various SALC lines that have interruptions in those genes. We could put on variants here. We could look at coverage of RNA-seq or gene expression. We could look at methylation. There's all kinds of features that we can put in here. So if you're maybe doing some genetic mapping and you identify a region of interest, maybe you're doing gene expression, you could say, okay, here's my genome of interest. It overlaps with this gene. Let me look at these insertional interruptions. So Genome Browser is a really great way to go from these regions of the genome into like discrete quantified genes and sequences that we care about. So in this notebook, we'll be playing around with Genome Browser a little bit. And so a lot of times you want to translate basic discoveries from models into crop species. And so the most important model in plants is this plant Arabidopsis. And so maybe you identify a gene that's important for disease or for stress tolerance or fruit development, or pick your favorite gene, pick your favorite trait in Arabidopsis. The question is, how do we find out what that equates to in other species? So maybe we want to find a gene involved in bacterial stress response in Arabidopsis, and maybe we want to know how that corresponds to pineapple or maize or strawberry or mango or walnut. And so we need to use comparative genomics to identify the genes that are in our model species and identify their corresponding orthologs or orthologous sequences in other species. And so there's a couple different ways that we can do this. And so generally plant genomes have conserved gene content, and so we call them collinear, meaning that the same genes or orthologous sequences are in the same relative order in the genome. And so throughout the last couple 100 million years, plants have undergone lots of rearrangements, losses, duplications, that sort of thing. But in general, a lot of genes tend to be kept in the same relative order respective to each other in genomes. And so what we can do is we can say, okay, we have this set of genes in our model organism, Arabidopsis, and then we can identify using Synteny, so using the sequence homology and the order across genomes, which genes are corresponding to other species. So we could say, okay, these 3 genes are in the same order and orientation in all of these species. Therefore, if this gene has this function in this species, it likely this is the orthologous sequence in this species. We could maybe say, here is this new gene in pineapple that's not found in any of these other species. Maybe it's important for some function. Maybe this gene is in Arabidopsis, but it's only in corn and not in the other species. And so Synteny refers to the order and orientation of genes along chromosomes and genes that have corresponding sequences. And so this is kind of an oversimplification in general, and this does not account for changes in polyploidy. So a lot of plant genomes are polyploid. They might have various structural rearrangements, gene losses, duplications, etc. So this is kind of an oversimplification, but generally it's a little bit more complicated than this. So my lab, the research in my lab focuses mostly on grasses, and so we do a lot of work with grass comparative genomics and stress tolerance in grasses. And so the grass family, or the Poaceae family, contains tons of really important, economically important species. So things like bamboos, rice, wheat, barley, rye, oat, sorghum, maize, sugarcane, various different types of millets, foxtail millet, pearl millet, teff, fonio, you name it. So the grass family contains tons of species of economic importance, and there's lots of research here. And so maybe you find a gene that's important in rice, and you want to see what the orthologous gene is in maize or maybe in teff. It could be involved in disease resistance, could be involved in stress tolerance. And so we can use comparative genomics to identify genes that correspond to each other. And so grasses are highly collinear. And so their genomes have about 80 to 90% of genes that are in the conserved order and in the same positions along chromosomes. And so here's an example showing a small region of the grass genome with this model grass Orpedium, foxtail millet, sorghum, rice, and another model Brachypodium. And so the genes are in blue and green with the colors denoting their orientation and the forward or reverse orientation. And then repetitive elements here are shown in orange. And then if a gene is conserved between species, it's connected by a gray line here. And so we could say that perhaps we're interested, we identify this gene in sorghum. We can use Synteny to say this gene corresponds to this gene in rice. It's in the same order, same orientation, and it has a lot of sequence homology. And that's the same gene in Brachypodium, or that's the same gene in Soteria and Orpedium. And so in general here you can see the vast majority of genes are conserved in the same order, the same orientation. There are some movements, so if we look here, this rice gene has moved in Brachypodium. If we can see that there have been some duplications and gene losses. So for instance, all these genes are novel in Brachypodium, these are novel in rice, and maybe there are some genes that are found in some species, but not in others. So I'm trying to see if there are any conserved ones here. I'm not identifying any. So we can use this to identify which genes are corresponding to each other and use this for like downstream functional analyses. And it makes it really nice to translate basic discoveries from cereals or from models to cereal crops or species in general. And so one complicating factor by this is that many wild and crop species are polyploid, meaning that they have more than two copies of each gene in the genome. And some great examples of this are, watermelon and banana are triploid, so they have 3 copies. Peanuts, blackberries are tetraploid, they have 4 copies. Things like wheat and sweet potato are hexaploid, so they have 6 copies. Strawberry and sugarcane have 8 copies. And there are species that have up to 12 or 20 or more copies of each chromosome. And so all plants have experienced at least one round of ancient polyploidy as well, or so an ancient whole genome duplication event, followed by, so this has a doubling of genes followed by a gene loss. And because of this, plant genomes, even though they do tend to have a lot of the same genes, they can be a little bit rearranged. They can be very complicated, and almost no plant genomes are truly one-to-one to each other. And so this complicates some comparative genomic analyses using Synteny. And so in the example I showed you with grasses, that's a little bit cleaner, but for other species, it's a little bit more difficult. And so here's an example with strawberry. And so the wild strawberry, one of the progenitors of octoploid strawberry, is diploid, and then cultivated strawberry is octoploid. So wild strawberry has 2 copies of every chromosome, and the octoploid has 8 copies. So I'm just showing you here 1 copy versus 4 copies. And so this is 1 copy of a region in the wild species, and this corresponds to 4 copies in the cultivated species. And again here in blue and green are genes. And so you can see the vast majority of genes are conserved in 4 copies. And so if we're doing downstream functional genomics, maybe we identify this 1 gene in wild strawberry that's important for a trait, but then we need to find the 4 copies that correspond in the polyploid species. And so polyploidy really complicates comparative genomics in plants. And this can be a massive problem for a lot of lineages. So Synteny is generally a good way to identify orthologous or conserved genes between species, but sometimes genes can move around the genome. And when there are new places, Synteny approaches are unable to identify orthologous genes. So if we have a gene that moves from one chromosome to another chromosome in one species, we might not be able to use its position to identify what genes it corresponds to. And so there's another approach that we can use. We can use a gene tree approach to identify conserved genes or orthologs across species. And so if we look at this phylogenetic-based approach here, let's say we have a singular gene here So this could be maybe a gene involved in disease resistance, maybe it's involved in some biochemical trait, whatever. We'll call this gene A. This gene gets duplicated in a genome, and then it turns into gene A and gene B. Maybe these two genes have different functions. And then we have a speciation event. So we could think of this like maybe here would be species 1 could be corn, species 2 could be sorghum. And then in each of these species, we have each species has a copy of gene A and a copy of gene B. And so the terminology we would use is that this gene A in species 1 and gene A in species 2 would be orthologs to each other because these are the same gene from a speciation event. And then we would say within a single species, gene A and gene B would be paralogs to each other because they can be traced back to the same duplication event, but that occurred before speciation. And we could have perhaps this other gene here that is unrelated to this. It has a different function, but a similar sequence homology, and we can call these analogs. But in general, we care most about the orthologs to each other. So we can use this phylogenetic-based approach to identify the origin of these genes, and then we can group them into genes called orthogroups based on their origin. And so there's a program that we use to identify orthologous genes across genomes, and the most common one that's used is called Orthofinder. And so this can be used to cluster any number of genes into orthogroups, or genes that are descended from a single gene in the last common ancestor of a clade of species. And so you could think of this like in, for example, disease resistance genes. There are hundreds of them in any plant genome, but they can all be traced back to one or a single or a couple ancestral copies. And so we can compare genes across species and then group them into genes based on their phylogeny, and then we can ascribe functions to different orthogroups. And so the way this works is we would have all the genes or a subset of the genes in each species. And so these could be like species A, B, and C. And maybe there are 30 to 100,000 genes in each of these genomes. We can use BLAST or a BLAST-like program to compare all the genes in each species against each other. So this is kind of computationally intensive, but this will basically show us which genes are similar to each other. We can group the genes by some cutoff into what we would call orthogroups. So these would be genes that have some homology to each other or some sequence composition that's conserved. We can then take these individual orthogroups and build a gene tree. So we can make a phylogenetic tree of how these genes compare to each other. And so for instance here we might say that if these genes here are from species A and species B, species B in green and species C in blue, like we would call this an ortholog to each other, and then these would be like paralogs. We can we can incorporate the species tree, we can reconcile the gene trees, and then we can have orthologs from this. And so we could say that this single copy in species A is orthologous to these two copies in species B and species C. And so from this we might get on the order of 10 to 50,000 orthogroups between any number of species, and then we can say, okay, here is one orthogroup from one species and see what this is involved in. So perhaps you have some orthogroups that are only found in some species and not found in others. You could functionally annotate those and say, what are these doing, and are these new genes that are involved in some lineage-specific traits? You can look to see if orthogroups have duplicated in one species over another. So maybe one genome has a bunch of orthologs involved in stress response that are duplicated, and that could be involved in like living in a harsh environment. Or you could say what genes have been lost. And so if you have like a parasitic plant or something, maybe it would lose specific orthogroups involved in photosynthesis or central metabolism. And so this is a really powerful way of, again, going from here are 30,000 random genes to here are groups of genes that are the same across species, and then we can do some comparisons to identify genes of interest or differences between species. And here's just to kind of give you one example. So this wasn't Orthofinder based, but it's something very similar. And this is just looking at these disease resistance genes or NBS-LLR genes in plants. And so these genes are responsible for various resistance to different pathogens. And this is just comparing the number of these genes across species. So like we could see here that this is orange, has the most of these genes, followed by tobacco and potato and Brassica rapa. And so we can identify genes, group them into ortho groups, and then do some downstream characterization of these genes. And so I don't know a lot about orange, but maybe orange has a bunch of these genes, maybe it's more stress tolerant, right? Or you could say, okay, I'm looking for this specific gene family, have all these genes here, and then you can do some downstream analyses. And so again, this is really a starting point, but it's a way to go from this species has 20,000, 200,000, whatever genes, and let's identify what's conserved and what's different between other species. And so just to summarize what we talked about today, so we talked about what makes up the plant genome and how genomes vary between species. So there are a lot of elements that are important in a plant genome. We still don't know what everything is doing or the role, or there's probably some unique biology that has yet to be discovered, but those are really important for different biological traits. We talked about how genomes in general are pretty similar to each other, plant genomes, that a lot of genes are conserved. They have the same orientation, the same order in chromosomes, and we can use that information to translate genes from one species to another. We talked about the different file formats that are used in genomics and what those mean and how we can use them to identify different features. And we talked about understanding foundational principles in comparative genomics. So two fundamental approaches that we can use to compare plant genomes. So using the Synteny-based approach of looking at where genes are in the genome with reference to each other and using that to assign orthology, or we can use a gene tree-based approach to identify orthogroups that are conserved between species. So this has hopefully provided you a nice introduction with how plant genomes are compared to each other and the downstream things that we can do to them. And so in the remainder of this lesson, we will be doing some comparative genomics analyses of our own, and then using a Synteny-based approach and an orthogroup-based approach.